Welcome to Manifested Publishers.
Welcome learners, my name is Stephen Kariungi. Today we continue with our discussion on osmosis, and specifically we are discussing water relations in animal cells. When you talk of the animal cells do experience osmosis. And that is when it comes to them absorbing water at different parts of the body. And it happens that this water is usually absorbed across the semi-permeable membrane, which is the cell membrane. And in this case, we are going to look at two scenarios. What happens when the amount of water in the body of the animal is less, and also when the amount of water in the body of the animal is high. So in this case, I will do that comparison. But before that, we can say that the cell membrane of animal cells is responsible for controlling the movement of substances in and out of the cell. It is semi-permeable. On this, we want to look at, for example, when animal cells are put in a hypotonic solution, and also when they are put in a hypertonic solution. So the first scenario is we'll take an example of red blood cells in a hypotonic solution. When the red blood cells are put in a hypotonic solution, they will definitely gain water. And they gain water by osmosis. Just like any other cell, put in a hypotonic solution, it gains water. And as the cell continues to gain water, it continues increasing in size, swelling, and eventually it gets to a point whereby that cell cannot expand any further. And at that point, the cell bursts. So we say that that particular cell has been hemolyzed or has undergone hemolysis. So in this case, we are saying that a red blood cell placed in a hypotonic solution, e.g. distilled water, gains water by osmosis, swells or increases in size, and eventually bursts. So the bursting of the red blood cell as a result of gaining water from a hypotonic solution is called hemolysis. So the process by which the red blood cell bursts as a result of gaining water from a hypotonic solution is called hemolysis. So we are saying that the cell continues gaining water until it cannot increase further in terms of the size, and therefore it bursts. So we say that that cell has become hemolyzed or has undergone hemolysis. Equally, when a red blood cell is placed in a concentrated solution or in a hypertonic solution, e.g. concentrated sugar solution, it loses water by osmosis and shrinks. So as a result of losing water, that shrinking is called crenation. This is called crenation. So we say that the cell has undergone crenation or has become crenated. So we have hemolysis and crenation. Those are two terms that we use in water relations in animal cells, whereby when the animal cells gain water by osmosis and burst, that is hemolysis. When animal cells lose water by osmosis and shrink, this is called crenation. We are going to illustrate that in form of a diagram. So we have, for instance, a red blood cell. And on this side, so this one is placed in a hypotonic solution. Just a moment. And on this other side, placed in a hypertonic. So the one that is placed in a hypotonic solution increases further in size. So first of all, it swells. And then finally, it bursts. Then bursts. And that's what we say becomes hemolyzed. So this process of bursting is hemolysis. On the other hand, if the same a red blood cell is placed in a hypertonic solution, it tends to shrink. So it shrinks and becomes crenated. So that process is what you are calling crenation. So the diagram shows the water relations in a red blood cell. So we can see from the first diagram, the red blood cell has a biconcave disc shape. When placed in a hypotonic solution, it gains water by osmosis and swells. That means increases in size. And then, if that continues, that particular cell bursts. It undergoes hemolysis. So we say that it becomes hemolyzed. And then when it is placed in a hypertonic solution, it loses water, shrinks, and undergoes crenation. So we can see how a crenated cell looks like. So we'll have an assignment on this. So the first question, what is hemolysis and what is crenation? Number 2, explain why a red blood cell bursts when placed in distilled water. So that marks the end of water relations in animal cells. We'll stop there. Next time we'll continue with the water relations in plant cells. See you next time.